Hi everyone, my name is Giulia and I'm a specialist in medical genetics. Today I would like to give you a very brief overview about DNA and to discuss a few applications of treating people with bespoke treatments to finding a suspect in a crime. So what is DNA and what is determining diversity among people? We all are different and each of us is unique. The only exception is made by identical twins that are identical and also their DNA is identical. So the reason why we are all diverse is that inside us we have DNA in which we store the information that makes us having being blonde, being brunette, or being tall or being short. And DNA is stored in our chromosomes that we can actually see in this picture. Chromosomes are within the nuclei of our cell. So in a cell we can distinguish a cytoplasm and a nucleus. So our chromosomes, which are made by condensation of our DNA, are within the nucleus of our cells. And the entire chromosomes, the entire number of our chromosomes, make what is called the human genome. So in all our cells, or in most of our cells, with the exception of our gametes, we have 46 chromosomes, 23 pairs of chromosomes. 22 pairs are called autosomes, and 2 instead are the chromosomes which make us becoming males or females, which are chromosome X and chromosome Y, which is instead specific for a male. So why are we so different? It is very difficult to believe, but we are almost identical in terms of sequence of DNA. So if we study our DNA, we see that despite us being extremely different, like these two people here, the basis, the content of the DNA is almost identical, because about 99.9% of their DNA is absolutely identical. Only about 1.1% of the DNA between very diverse people like these two people here, or all the people in this other slide, is actually making the difference. So it's explaining why this person is very short, why this person instead is very tall. So the information about what we look like is stored in our DNA. DNA stands for deoxyribonucleic acid and has a structure of a double helix that looks like a twisted ladder. The identification of the structure, this fantastic structure of the DNA, was made in 1953 by 2 scientists, which were Watson and Crick, which were then awarded with the Nobel Prize. What is the function of the DNA? The DNA contains our genes, so segments of DNA, many segments of DNA, code for our genes, which are the ones that then will make our eyes be blue or our hair be blonde, for example. So the sequence of all these genes on the DNA will then be compacted and stored in our chromosomes, in our nuclei. So the DNA, if we analyze the structure of the DNA, we will see that it's made of 2 strands, which then bind together and form this double helix, so they're twisted together. And this strand is made of repeating subunits, which are called the nucleotides. A nucleotide is a structure which is composed by 3 elements. We can recognize a phosphate group. There is a sugar, which in the case of the DNA is the deoxyribose. And then there is a base. In the DNA we have 4 possibilities. So we have adenine, thymine, guanine and cytosine. So these bases are actually capable of binding to each other, but in a very specific way. So there is what is called a base pair rules. So adenine is only able to bind to thymine and guanine is only able to bind to cytosine. So the reason why the DNA forms this double helix is that all these bases which are linked to each other together then can recognize the complementary base, and so they can make these bindings, which are actually hydrogen bonds, which make these strands staying together but not bound in a very, very strong way. So there is a rule, that is the base pair rules, for which adenine can only bind to thymine and cytosine can only bind to guanine. So if I know what's the sequence of one strand, I can then predict what is the sequence of the complementary strand based on the complementarity between bases, because A can only bind to T and C can only bind to G. So DNA contains our information, but what happens in our cells is that the DNA contains the genes, but then we need to make the proteins, which are the blocks that are making us moving, for example, that make our muscles. So for us to pass the information from the DNA to proteins, the DNA must be first transcribed into a molecule which is called the RNA, and then the RNA must be then translated into proteins, which are actually the blocks. And the frame, the code that is able to read and to translate the bases into amino acids, which are the blocks which compose the proteins, is called the genetic code. So we have our DNA, which is in our nucleus, then the DNA is transcribed into a molecule of RNA, and then using amino acids, the RNA is then translated into a protein, and proteins are the building blocks of our entire organism. So what happens if there is a mistake in our DNA? As I mentioned, we need to use codons, triplets. So as you can see here, I have triplets CAC, CTG, ACT, and so on. And each of these, according to the genetic code, will correspond to a specific amino acid. So what happens if by mistake I change even one of these nucleotides, one of these bases? For example, I change from GTG to GAG. Even though we can think that it's a tiny mistake because I'm only changing one nucleotide, the effect I can have can be very serious, because I may change from one amino acid, which is the glutamine, to another amino acid, which is valine. And this can be very important, because even one single amino acid can be responsible for destroying the function of a protein. So if I have even one tiny mutation in my DNA, I can end up having a disease. This particular mutation, for example, is specific for a disease which is called sickle cell anemia, in which our red blood cells don't have the normal disc shape, but instead look like sickle, and we can see these abnormal red blood cells in a blood smear. Because of this, they cannot transport oxygen very well, and they can also block the microcirculation, so we might have a very serious disease because of this very tiny mistake in our DNA. So it is important to bear in mind that there are some diseases which are called genetic diseases, because they are caused by mistakes in our DNA. Sometimes these mistakes are very tiny, so can be mistakes of one single base, as in the case of sickle cell anemia that I've just shown. Sometimes instead I can have the change of one chromosome, or even more than one chromosome, even the entire set of chromosomes. Obviously, the bigger is the mistake, the bigger will be the effect in terms of seriousness of the disease. So it is important that we try to understand when we have a patient and we think that this patient has a genetic disease, what is the particular mutation, what is the particular mistake that is in the DNA of this person, because when we know the mistake, we can possibly give the best treatment. So there are many techniques that we can use to diagnose a genetic disease, and the technique that we will use will depend on how big is the size of the mistake in terms of length on the DNA. So we can identify very big mistakes, so big pieces on a chromosomes that are lost or gained using a technique which is called karyotyping. We can have the possibility of identifying smaller gains or losses, so deletions or gains, by using another technique which is called, for example, array CGH, which is shown in this picture. But what I would like to discuss with you instead today is how we diagnose very small, tiny mutations, and so why we use DNA sequencing. And in particular, I will briefly mention only one technique, which is the Sanger sequencing. So this is an example of a Sanger sequencing. So how do we read the composition of the DNA? So we make the DNA sequencing, and we literally read the sequence of the DNA. So what are the letters that we find? A, T, C, G, and so on. But before we discuss how we can sequence the DNA, can look at how the DNA is built in a cell. As I mentioned, the DNA is made of two strands, and these strands are complementary to each other because they are made by these blocks, by A, T, C, and G, and we know that A can only bind to C and C can only bind to C. So when I have to synthesize the complementary strand of the DNA, if here in this position I have an A, then here I will have necessarily a T. If here I have a C, here I will have necessarily an G, and so on. So I will have what is called an elongation of the complementary strand. This scientist, which is Sanger, who was awarded in 1980 with a Nobel Prize for his this discovery, introduced a particular way of sequencing the DNA, which is an enzymatic method, which is called the dideoxy procedure. So he understood that we can block the elongation, the growth of the complementary strand, if instead of giving a normal nucleotide, we give a defective nucleotide in which this particular group, which is called an oxidryl, this made by oxygen and hydrogen, is then substituted, replaced by the hydrogen alone. Because of this, if we incorporate into our strand of DNA this defective nucleotide, the DNA, the elongation of DNA cannot go further any longer, and so the DNA will be stopped. So if we are using a mixture of nucleotides containing also some defective nucleotides, which are the dideoxy nucleotides, at some point and randomly we will stop the synthesis of the DNA. And if this dideoxy nucleotide, this defective nucleotide, are bound to either a radioactive molecule or a fluorescent dye, we will be able to identify these different fragments of the DNA. So I will show you two different examples of how we can identify a sequence of the DNA. The first one is binding, so having a radioactive molecule bound to these defective nucleotides. This is a method that was very important at the beginning, but now is not any longer used because it's obviously dangerous. And now instead, these defective nucleotides are bound to fluorescent dyes. So when we incorporate these defective nucleotides, these particular molecules of DNA whose synthesis has been stopped, whose elongation, whose growth has been stopped, will emit a particular light if excited. And so we will have a graph in the end of the reaction in which we will have a lot of peaks, and each of these peaks and each of these colors will correspond to a specific base. So when we will then look at the signal, we will know that, for example, Na will be given by a green peak, a G will be given by a indicated by a black peak, a C will be given by a blue peak, and instead a T will be indicated by the presence of a red peak. So if we just look at the colors, here we can actually, based on the correspondence with between a base and a color, we can actually here give the sequence of the DNA. So here I have 3 peaks which are all red, so I know that red corresponds to T. So here I know that I have T, T, T. Then I have 3 green peaks, and I know that the green peak corresponds to an A. So I know that after T, 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 I have A, A, A. Then I have a blue peak, which is AC, so I have C. And then I have 3 black peaks, which are the G's, and so I have G, G, G. And so like this, I can actually continue. So for example, if we look at this slide here, we see that I have an A, I have a G, and then here I have 2 overlapping peaks because I can see 2 signals. I can see a red signal and I can see a green signal. So these overlapping peaks means that I have identified in this particular point a mistake in my DNA. So if I identify a mistake, I can possibly find a possible cure, a possible treatment. So why do I want to sequence my DNA? Because as I mentioned, the more I know my DNA, the more I know that I can give bespoke treatment to people. And because each of us is different, if we receive bespoke treatments, we will have less side effects and we'll have a better outcome. So we will have what is called a personalized medicine. So not a drug that is for everybody, but drugs that work on each of us, individual people. Let's talk about what is called a bespoke drug. And as an example, I will talk about a cancer, which is a leukemia. In this leukemia, because there is an exchange of pieces between two chromosomes, which is called a translocation, two genes that are normally kept separate, which are the gene BCR and the gene ABL, are put together, are then fused together, and this generates an abnormal fusion protein, which is called the BCR-ABL. Why is this a problem? Because this protein that is now fused codes for a receptor for a protein that cannot be switched off anymore, and so keeps working in a cell, sending constantly a wrong signal. So for these people to be cured, we need to switch off this protein. So we know that in these people we have this fusion protein. So there are some particular drugs that can be able to bind to this protein and to switch it off. So if I give to only these particular people that have this particular fusion protein, these drugs, these people will be cured. Obviously, this drug will not work on any other people that will not present with this particular fusion protein. This is why this is a bespoke drug, because it only works for people carrying this fusion protein, the BCR-ABL. Another example can be offered by the use of another drug, which is tamoxifen, another drug used in cancer, especially in a particular type of breast cancer patients. This drug is usually given in an inactive form and then is activated to kill tumor cells in our liver cells in our body. And the activation, the activation of this drug is due to an enzyme which is called CYP2D6. Now, for some reasons, it was observed that about 7 to 10% of women who were receiving this drug were not responding. So even though they were presenting with exactly the same kind of cancer, they could not be cured by this drug. So it was understood, it was analyzed, the sequence of this enzyme, and it was seen that in these particular women who do not respond to this drug, there are some differences in terms of the DNA. The enzyme is actually not working. So if we know the sequence of this gene in these women with breast cancer, we can avoid to give them tamoxifen, because we already know that tamoxifen will not work, and we will instead give them an alternative drug. Knowing the sequence of a DNA can also be very useful to predict how likely it is that we will get a disease. And this is especially true for some particular familial diseases, like, for example, breast cancers, which are very, very frequent in a particular family. We know that breast cancer can be due to mutations of two genes, which are called BRCA1 and BRCA2. So there is a mistake in the DNA coding for these particular genes. And because of these mistakes, people become very likely to develop, very susceptible to develop breast cancer. So what happens if we know that these people will have a mutation in this gene? So if we read the sequence of the DNA of these people and we identify a mistake in their DNA, we will know that their likelihood of developing breast cancer will be much higher than the likelihood of developing breast cancer for a person in which these genes are normal, in which the sequence of these genes is not mutated. So knowing the likelihood, knowing that these people are more likely to develop breast cancer will be very helpful, because they will be monitored very often. So we can prevent them from getting cancer, or we can treat these patients when the cancer is very, very small. So the prognosis will be much better. But I want also to say that knowing the sequence of the DNA is not only useful in medicine, although that the applications in medicine are endless, I would say, because DNA is also very useful in forensics. For example, let's imagine that we are on a crime scene, we found some biological traces, but then there are many suspects, and so we cannot know which one actually did the crime. So we can simply take a swab from the mouth of these people and extract DNA from each of the suspects, and then we can compare the traces that we found on the crime scene with the DNA from our suspects. And in this particular cartoon here, we can see that the DNA found on the crime scene corresponds perfectly to the suspect number 2. So because their DNA match, their DNAs match, we can then see that the crime was actually made by suspect 2. So we found the culprit, and we can also exclude other suspects which are totally innocent. So application in forensics of the sequencing of the DNA are also extremely useful and extremely important. But there is also another thing that can be of interest. For example, we might be interested in finding our ancestors. So if we analyze DNA in our families, we can go back through generations, and we can identify, we can understand where we are coming from, who were our ancestors, which is another very useful and interesting application of the DNA. So I will stop here. I could actually continue because there are many other interesting things that we can do with our DNA, and I really hope that this very short talk stimulated your curiosity, and you will want to know a bit more about DNA and what we can do with the DNA. And I hope many of you will then become scientists and maybe hopefully geneticists. Have a nice time and good luck for your future.